Okay, 3.6.2.1 nerve impulses. We're going to quickly talk about the structure of nerves, and then next lesson we're going to move on to look at the generation of the action potential. But we really can't do that until we have this sort of fundamental knowledge to begin with. So all messages that are sent in the body are generally split into two categories. We have local mediators as well, which is something different, but we have hormones, the endocrine system, and we have the nervous system. So hormones send chemical signals as opposed to electrical signals in the nervous system. They are long-lasting as opposed to short-lasting, and they generally are very slow because they're moving around in the blood. They take a little while to get to the receptor and make a change and make an effect, whereas nervous impulses are very fast. So a nervous impulse is an electrical impulse, and what we mean there, that we'll look at more next lesson, is a change in membrane potential. So membrane potential in physics is measured as volts. We're talking about very small voltages here, so we measure the membrane potential in millivolts. So we'll look at the term depolarization and the change in positive membrane potential on the axon membrane next lesson. So here's the general structure. So what we're looking at here is a motor neuron. So a motor neuron will connect to an effector, a muscle or a gland at this end, and what will happen here is that the impulse will move in this direction, will move down, and will stimulate the muscle to be contracted. So we've got dendrites at this end, which are extensions from dendrons that come out the end of the axon. We have the cell body up here, and this has its own dendrites that are going to connect to an appropriate relay neuron. Inside the cell body, we have the nucleus, and then we have the axon. Now, what we're looking at here is an unmyelinated axon. We'll talk about what that means in a second. So sensory neurons are a little bit different in that the cell body is in the middle of the axon, and it doesn't have any dendrites. So here is the axon. At either end, we have dendrons, and coming off the dendrons, we have dendrites. So remember, sensory neurons connect a receptor to a relay neuron, unlike the motor neuron. And then finally, if we look at the relay neuron, that really doesn't have any axons at all. It has this cell body in the center, and then it has dendrites and dendrons that are coming from it, and they will make multiple connections between sensory neurons and the motor neuron. Remember, we find that in the central nervous system, the spinal cord. So we go back to what we're looking at here. There's a few other kind of key sections we need to know, in that wrapping around the axon, we have specialized cells called Schwann cells. And these Schwann cells, within their cell membranes, have a fat called myelin. That myelin insulates the axon. So if you imagine the Schwann cell kind of wrapping around, it creates these layers of myelin within the layer. And that means that we've got these lumps like this of deposits of myelin as a result of being inside the wraps of Schwann cells. And that creates junctions in between the Schwann cells. We call these spaces here, these gaps where there is no myelin as a result of being known Schwann cells, they're called the nodes of Ranvier. And they're basically spaces where there is no insulation. And that's going to allow the action potential, the electrical impulse, to be propagated a bit faster than it would be if the axon is unmyelinated. So here we're looking at an axon that has no myelin on it, and therefore is unmyelinated. So generally, when we're talking about axons having a myelinated structure, that happens to speed up conduction, as we'll see in a minute. Now, it also prevents impulses from jumping from one nerve to another, because if this becomes positively charged, potentially some of that electrical impulse could initiate a response in another axonal membrane if it was very close to it. So this insulation also prevents cross conduction, which is also to be considered, not just the speed of transduction.